important phagocytic cells. So remember, in endocytosis, the cell membrane invaginates and forms vesicles to isolate foreign particles. And endocytosis is the mechanism for viruses to enter cells. For number 10, the answer, exception po ang letter D. Okay, exception po or incorrect po ang letter D. Number 11, the infectious disease process in which large numbers of activated macrophages and histiocytes are collected at the site of inflammation. This is called as letter A, purulent, B, granulomatous, C, chronic, letter D, opportunistic. The answer for number 11 is granulomatous. This is a description of granulomatous. If many activated macrophages and histiocytes aggregates in the area, okay, in an injured area, particularly with the formation of multinucleated giant cells, the reaction is called granulomatous. Okay, the answer here is letter B, granulomatous. The sequence of an inflammatory reaction begins with a local accumulation of segmenters. We call this as your segmented neutrophil. Okay, your segmenters. Okay, or neutrophil. Okay, so these neutrophils, okay, will be at the injury site. Okay, a local accumulation of the cells. Okay, the stage is called the purulent or suppurative stage. Okay, so if the infective agent is not removed by these neutrophils at the purulent stage, okay, this will followed by the mononuclear. Okay, the mononuclear inflammatory cells, okay, will now invade the area, and this will lead to chronic infection. Okay, this will lead to chronic infection. This mononuclear inflammatory cells includes lymphocytes and monocytes, and of course macrophages. So plasma cells are often plasma cells become an integral component of the chronic inflammation, okay, the chronic inflammatory reaction, when the antibody formation is at the most, okay, production or at its peak. Opportunistic infection, this is term used to describe the evolution of an infectious disease in an immunocompromised individual by bacterial species, okay, or other pathogenic species that does not commonly cause infection in healthy host. Okay, in a healthy host. So number 11, again, letter B is the correct answer, granulomatous. Number 12, compared to the primary immune response, the secondary immune response typically demonstrate which of the following? A, less antibodies produced. B, a longer lag time. C, more antibodies produced. Or letter D, lower affinity. So for here, secondary immune response, this is typically presents with a greater number of antibodies that are produced as compared to the primary immune response. So in this case, the answer is letter C. So the primary immune response typically presents with the generation of less antibodies as compared to secondary immune response. The secondary immune response presents with a shorter time, okay, as compared to the primary immune response. Okay, secondary immune response typically presents with antibodies that have higher affinity as compared to those from primary immune response. So that's the difference between the primary and your secondary immune response. Number 12, letter C. Number 12, it is letter C. So to summarize, number 12, more antibodies produced in the secondary immune response. Number 13. The recognition unit of the classical complement pathway refers to which of the following? Letter A, C5b, C6, C7, C8, C9, B, C1q, C, C3a, or letter D, C4. For number 13, as described in classical complement pathway, the recognition unit is C1q. So C1q, kanina nasabi natin siya, C1q, this is the term or referred to as the recognition unit. Okay, it refers to the recognition unit of the classical pathway. So C1q is the molecule which bind to the antibody, particularly at its Fc portion. Okay? So, C5b, C6, C7, C8, and C9, it makes up the membrane attack complex or the MAC. Okay? So, this is your C5b, C6, C7, C8, and C9, which makes up the membrane attack complex. So, cell lysis through the formation of the MAC or the membrane attack complex consisting your C5 to C9. Okay, the C5, C6, C7 complex acts on the C8 and C9. This C8 and C9 are inserted into the cell membrane, which will later on lead to the formation of lesions, okay, that will allow passage of ions, and definitely later it will result to cell lysis caused by osmotic pressure changes. Okay, we have your C3A. C3A, it works as a natural substrate on C4B, 2A. Okay, your C3A, it acts as a natural substrate for C4B, C4B2A, while your C4, along with C2, okay, this is part of the classical complement pathway. So, number 13, the answer is, for a recognition unit, this is your C1q. Okay, recognition unit. Okay, number 14. The purpose of your C3A and C5A, okay, the split products of complement cascades is to, letter A, bind with specific membrane receptors of lymphocytes and cause release of cytotoxic substances. Letter B, cause increased by a vascular permeability, contraction of smooth muscle, release of histamine from basophil. Letter C, bind with membrane receptors of macrophage to facilitate phagocytosis and the removal of debris and foreign substances. Or letter D, regulate and degrade membrane cofactor protein after activation by C3 convertase. The answer for number 14, C3A and C5A, is to letter B, cause increased vascular permeability, contraction of smooth muscle, release of histamine from the basophil. So C3A and C5A, okay, split products of the complement cascades. These are the split products of your complement, okay, cascade. Okay, their purpose is to cause increased vascular permeability. Okay, they should cause, okay, increased vascular permeability, contraction of smooth muscle, and release of histamine from basophil. C3A and C5A, bioactive fragments of your complement components C3, protein C3 and C5 respectively. They play a key role in the mediation of immunologically activated or provoke inflammatory responses. So C3A and C5A are produced by the activation of the classical pathway and or the alternative pathway. So this components, these are considered as your anaphylatoxins. Or these anaphylatoxins, will cause the basophils as well as the mast cells to release histamine, causing the smooth muscle contraction and again, vascular permeability. Okay? So, among dun sa tatlo ay sa ating mga anaphylatoxins, including also C4A also, okay, C4A, these are anaphylatoxin, C3A, C4A, and C5A. Ang tanong, kapag tinanong, which of the following daw, okay, is the most potent? This is C5A. Okay, most potent, okay, would be C5A. This is followed by C3, followed by C3, followed by C4A. So, pagka-potency naman ng pinag-uusapan, C5A is the most effective, followed by your C3A, followed by your C4A. Number 14, the answer is letter B. Caused increased vascular permeability, contraction of smooth muscle, and release of histamine from basophils. We have number 15, the late steps of complement activation and formation of the membrane attack complex results in, kanina na-describe na natin siya, ano? So, letter A, immune complex removal. B, lysis of cells. C, opsonization. Okay, in phagocytosis. Or letter D, polymorphonuclear leukocyte activation. Okay, so kanina nasabi natin that if once the MAC is formed, okay, from the components of your C5 to C9, okay, this will cause the production or the formation of cell lesions. So these cell lesions, butas, okay, will allow rapid passage of ions. And this passage of ions, okay, will cause changes in this cellular osmotic pressure, okay, leading to cell lysis. Puputok siya. Okay? So, let's just discuss letter B, ha? So, lysis of cell, okay, this is a result of the late steps of your complement activation and the formation of your MAC. So, once it is fully assembled in the correct proportions, okay, this will cause, again, okay, lesions. Okay, this will cause lesions. Sometimes they term this as tubule or pores, okay, which spans to the membrane of the cell wall, okay, that is being attacked by your complement. Okay, so the ions will flow freely, okay, between the cellular interior and exterior structures. Okay, and this will cause, okay, osmotic cytolysis. Okay, cytolysis. Okay. Okay, let's discuss the other choices. We have the letter A, immune complex removal. So, removal of an immune complex from tissues occur from the activation of your classic complement components, C1q, excuse me, and covalently bonded fragments of C3 and C4. That is letter A. Opsonization in phagocytosis, this is facilitated by covalently bonded fragments of C3, okay, C3 and C4. Okay, so covalently bonded fragments of C3 and C4. So that is letter C. D, polymorphonuclear leukocyte activation. This is facilitated by, okay, by C5A, C3A, and your C4A. Okay, polymorphonuclear leukocyte activation. This is facilitated by your C5, C3A, C4A, okay, components. Number 16. The major histocompatibility complex or MHC, okay, one genes encode for letter A, okay, human leukocyte antigen, A, B, C. Letter B, HLA-DR, DQ, DP. Letter C, complement. Letter D, okay, cytokines. Okay, so for this question, the answer is letter A. MHC, okay, 1 genes or class 1 genes, this will code for the antigens of the HLA A, HLA B, HLA C. Letter A. So, remember that your human leukocyte antigens, these are cell surface markers. Okay, these are cell, okay, surface markers. So, these markers allow the differentiation of self from non-self. So, these antigens were first described among WBCs, okay, and are coded by the genes in the MHC. So this MHC gene is located on the chromosome 6. So this gene is responsible in the following product classes. So class 1, we have your HLA, HLB, HLC, HLE, HLF, HLG, etc. So this molecules are found on nearly every nucleated cell surface. Very important itong class 1 that we have to remember that those molecules or those cells or antigen-presenting cells containing MHC1 molecules, okay, this present antigen to your CD8 positive cytotoxic lymphocytes. So again, those antigen presenting cells with MHC1 molecules, they present to your CD8 positive CTLs or cytotoxic lymphocytes. Okay, while in your class 2, okay, while in your class 2, these are your products, your HLA-DM, HLA-DO, DP, DQ-DR. So these molecules are located on the surfaces of the monocytes, macrophage, B cells, activated T cells, dendritic cells, Langerhans cells, and some epithelial cells. So important to remember also in the class 2 molecules. So this class 2 molecules containing antigen presenting cells. So antigen presenting cells with MHC 2 molecules, they present antigen to T helper cells. Again, so those antigen presenting cells with MHC class 2 molecules, they present, okay, antigens to the CD4 positive T cells. So CD4 positive T cells, these are our T helper cells. Okay? On the other hand, we have your class 3 products, MHC products. So these are the complement proteins, the TNF alpha, TNF beta, okay, and other proteins. Okay, this and other proteins, okay, that are not associated with cell membrane surfaces. Okay, not associated with cell membrane surfaces. So in this case, letter A, these are your products or antigens or molecules, okay, from your class 1, okay, class 1, considered as your class 1, Okay, MHC class 1 products. HLA-DR, DQ-DP, class 2 ito. Complement. Okay, these are complement and cytokines. These are part of the class 3, okay, MHC products. Number 16, letter A. Number 17. Number 17, the prozone effect can be described by all of the following except letter A, result in a false negative reaction. B, the result of antibody excess. Letter C, dilution of antibody can help prevent its occurrence. Or letter D, results in a false positive reaction. So all are correct. Okay, all describes prozone, okay, except letter D. Okay, so prozone does not cause a positive reaction, but rather a false negative reaction. So, tandaan natin ang prozone effect, okay, it is a result of antibody excess. Okay, antibody excess. That is your prozone. Okay, ang kontra niyan, we have your postzone. Okay, your post zone. Okay, dito naman, instead of excess tayo ng antibody, we have excess of antigen. Okay, so balik tayo with prozone. So, prozone, okay, the concentration of antibody exceeds the concentration of the antigen. So, therefore, it appears as a false negative reaction, which can become positive, okay, if the patient's serum is diluted. Okay, so remember, okay, prozone reaction as well as your postzone reaction does not cause or does not yield a false positive reaction. Okay, so hindi po yan. Nung student po ako, para po medyo maalala ko po siya, yung prozone, di ba po pagka sinusulat siya, mukha siyang antibody. Okay, sa prozone. So, antibody excess. Tapos, yung postzone, mukha siyang bilog. Mukha siyang antigen, no? So, postzone. So, antigen excess. So, para lang po hindi makalimutan, no? Okay. So, number 18. Which one of the following statements is false? Letter A, fever can have a direct effect on the growth or death of pathogenic microorganism. Letter B, low pH of stomach, skin, vagina can inhibit microbial growth. Letter C, secretory cells of the innate immune system are antigen specific. Or letter D, oxygen tension can result in the death of microorganism. Okay? So, all of the following are correctly stated except letter C. Okay, so secretory cells of innate immune system are antigen specific. No, mali ito, no? So number 18 is incorrect. Why? So remember that innate immunity system or innate immune system, it is inherent, okay, and non-specific. Okay, it is inherent and non-specific. So meaning, all pathogens are attacked, okay, similarly and are not antigen specific. Okay, so that is the innate immune system. Okay, hindi siya antigen specific. Okay, so fever will have a direct effect on the pathogen, okay, on the microorganism, and therefore it can inhibit the growth of the microorganism and will later on cause death. pH has a direct effect on the pathogens, limiting their survival or their ability to produce or to reproduce. And we have your oxygen tension. Oxygen tension, on the other hand, has a direct effect on many pathogens, okay, resulting to death due to oxidation. Okay, due to oxidation. Okay? So again, fever, okay, the increase in temperature will prevent growth, and therefore, it will cause death. pH, okay, has a direct effect, okay? So, limiting the survival, okay, limiting the survival and the ability to reproduce. Oxygen tension, okay, it will cause death, okay, due to oxidation. Okay? So, number 18 po, C is incorrect or falsely stated. Number 19, which of the following antibody types is chiefly seen in the primary immune response? Letter A, IgG, B, IgA, letter C, IgM, letter D, IgD. So, kanina na-discuss natin to, primary immune response, ano? So, this is involved with IgM. So, IgM is the main immunoglobulin, okay, seen in the primary immune response, okay, which this reaction or this response occurs after the first exposure to an antigen. Okay, so present after the first exposure to an antigen. While your IgG is chiefly seen, okay, okay, in your secondary immune response or memory response, okay, and this is produced upon second or subsequent exposure to an antigen. Okay, so it is produced okay. So IgA is predominant immunoglobulin in D. Okay, so this is the immunoglobulin immunoglobulin in the secretions. Okay, in the secretions. Finally, we have your IgD. IgD is found in small quantities and accounts for less than 1% of the total immunoglobulin pool. Okay? So, number 19, the answer po for the antibody that is chiefly seen in a primary immune response, we have your IgM. Okay? IgM, letter C. Number 20. Which brand of the immune system has an immediate response on the first exposure to a foreign antigenic stimulus? Again, which branch of immune system has an immediate response on first exposure to a foreign antigenic stimulus? So, choices are A, cell-mediated, B, specific, C, humoral, D, innate. So, as discussed kanina, this is described as the innate immunity. Okay, exposure to a foreign antigenic stimulus. So, innate immunity, inherent, non-specific. So, definitely lahat ng microorganism, okay, they will be attacked, okay, in a similar way, okay, because this type of immune response is non-specific. Okay. So, example ng innate immunity, kanina, we have your external defense mechanism, the skin, mucus, respiratory tract, acid pH of the stomach, of the skin, okay? And cell-mediated and humoral immunity, or responses, cell-mediated and humoral responses, they, the two main arms of specific or adaptive immunity. So, these are part of your adaptive immunity. So, adaptive cell-mediated, okay, adaptive humoral, okay, mediated. Okay, so these responses take a longer time than the innate defense to be initiated, okay, because it takes time for the T and B cells, okay, or T and B lymphocytes to become specifically activated against the foreign antigen. So, number 20, the answer is letter D. 21. Phagocytosis by cells of the mononuclear phagocytic system is greatly enhanced by which of the following? Letter A, hemolysin, B, opsonins, C, specific antitoxins, or letter D, neutralizing antibodies. So let's define first the given terms. We have letter A, hemolysin. Hemolysin, these are substances such as your streptolysin O. As we can recall, streptolysin O, these are produced by most group A streptococci. So this group A streptococci will produce streptolysin O, okay, that will cause disruption of the membrane integrity of the red blood cells. Okay, once membrane integrity of the red blood cells is disrupted, okay, this will cause the release of hemoglobin. Okay, we call that as the hemolysis. Opsonin. Okay, opsonin is actually the correct answer in number 21. Okay, so an opsonin, is a chemical substance that binds to antigens, okay, increasing the rate and quality of action by the phagocytes, okay, to destroy invading microorganism or particles. So this process is what we term as opsonization. Okay, in opsonization, the complement component C3b, okay, is attached to a particle, okay, okay, which promotes the adherence of the phagocytic cells, okay, because of the C3 receptors. So C3b attached to the particle, okay, promotes phagocyte adherence. Okay, promotes phagocyte adherence. Okay, and therefore, okay, phagocytic action will commence. So antibody, if in case, okay, present, it will augment this process by binding to the Fc receptors. Okay. Next, we have your specific antitoxin. So antitoxins is our antibodies, okay, that interlocks or bind with and inactivates the toxin. These toxins are a poisonous substance, can be in a poisonous substance. Okay? And this toxin could be produced by certain bacteria. Okay? We have letter D, neutralizing antibodies. Antibodies that defend or neutralizes the biological effect of cells from an antigen or infectious microorganism. How? By inhibiting or neutralizing any of its effect. So for number 21, again, the answer po is letter B. Okay, letter B, opsonins. Number 22. Which of the following statements concerning phagocytosis is false? Letter A, cells are capable of phagocytizing bacteria. B, membrane reaches out and surrounds the material to be internalized. C, once internalized, the material is contained in a structure termed as phagosome. Or letter D, only specialized cells are capable of phagocytosis. So, remember, phagocytosis, this is carried out by specialized cells of the innate defense system. Mainly, we have the neutrophils, we have monocytes, we have macrophage. These are the most common examples of phagocytes. So this blood cells, these white blood cells do not only phagocytize your bacteria, okay, but they can also phagocytize other cells. Okay, other cells, including also the remnants of previous phagocytosis, waste products. Okay, so after contacting the material to be internalized, the phagocytic cell extends its cytoplasmic membrane to surround the material. So, parang Pac-Man, no? Parang kakainin niya. Okay? Lalakihan niya yung bunganga niya, tapos kakainin niya siya. Okay? So, these phagocytes, okay, will surround the material and will later be engulfed by this phagocyte. This structure formed, okay, the structure formed, okay, okay, the structure formed here, okay, the structure formed here, we call this as your phagosomes. Okay, your phagosome. So, given, kunwari, ayan, okay, nag-i-engulf siya ng isang bacteria, for example, okay, so this will be engulf, okay, and the structure that is engulfing the bacteria, we term this structure as your phagosome. Okay, so lysosomal enzymes from the phagocytic cell, okay, are then released into the phagosome. Okay, what will happen? So maglalabas siya ng lysosomal enzymes, okay, and this will destroy or digest, okay, the engulfed material. Okay, so cells are only capable of phagocytizing bacteria. This is mali. Okay, they should be a specialized, okay, specialized cells capable of phagocytosis. Okay. B, membrane reaches out and surrounds the material to be internalized. Correct. C, once internalized, the material is contained in a structure termed as your phagosome. Letter D, specialized cells are capable of phagocytosis. And the following are examples. So what is incorrect here is letter A. So this is from 3rd John, showing the 6 processes of phagocytosis. First, of course, chemotaxis. Okay. Number 2, adherence. Number 3, engulfment. 4, the lysosome. Okay, and B, fusion of the organelle in the bacterium. And number 6 is the digestion and destruction. Number 23, the main purpose of neutrophil granules is to, number 23, primary purpose of neutrophil granules is to, letter A, facilitate nuclear maturation. B, distinguish neutrophils from lymphocytes. C, prepare cells for the removal from circulation. Or letter D, provide microbicidal actions. Okay, so neutrophils, again, these are phagocytes. So these are key component of the natural or innate defense systems. So neutrophil contains granules, okay, and these granules contains enzymes. Okay, so this contains enzymes. So these enzymes are needed, okay, or function to break down substrates. Okay, to break down substrates. Okay, therefore, killing the bacteria. Okay, killing the ingested pathogen. Okay, and secrete other contents during an inflammatory response. So this enzyme is needed to destroy or break down substrates, okay, killing the ingested pathogen, and okay, these are needed or secreted, okay, to induce inflammatory response. Okay, response. Okay, so to induce inflammatory response. Response. Okay, so the granules of neutrophils do not have role on nuclear maturation or in preparing cells for the removal, okay, from the circulation. Okay, while granules are a feature that helps distinguish neutrophil from lymphocytes, this is not its primary purpose. Okay, not a primary purpose. So, hindi si A, hindi rin si B, and C, hindi. So, the answer is letter D. Okay, provide microbicidal action. Okay? So, ito. So, number 24, antigen processing, okay, is primarily accomplished by which types of cells? Letter A, basophils, B, eosinophils, C, PMNs or polymorphonuclear neutrophils, D, macrophages. So, let's define first the antigen presenting cells. Okay, what is the antigen processing? So, in antigen processing, you need antigen presenting cells, or we term as your APCs. Your APCs are group of functionally defined cells, okay, capable of taking up antigens. Okay, so these antigens will later be presented, okay, presenting them to the lymphocytes. So these are cells capable of taking up antigens and presenting them to the lymphocytes. So this APCs includes your macrophages, dendritic cells, B cells, and even tissue cells. Okay, and also tissue cells. Okay, but pinaka-common, okay, APCs, we have your macrophage, dendritic cells, and your B cells. Okay, so we have your macrophages. So your macrophage, okay, your macrophage along with the network of reticular cells of the spleen, thymus, and other lymphoid tissues, okay, these are organized into the mononuclear phagocyte system. Okay, so we have specialized macrophage depending on the site or location or organ. So we have the macrophage, okay, located in the liver. These are the Kupffer cells. Kupffer cells are specialized macrophages located in the liver, lining walls of the sinusoids that form part of mononuclear phagocyte system. So functionally, monocyte macrophage, okay, have phagocytosis as their major role. So major role of this monocyte macrophage system, they are phagocytes. Okay, they do phagocytosis. That's their major role. But these cells perform at least 3 distinct but interrelated function in the host cells defense in host defense. So ito yung itong mga to, the monocyte, okay, macrophages, okay, ang main function daw niya is to do phagocytosis. So, the phagocytic property of the macrophage is particularly important in processing the antigens as part of the immune system. So, after the phagocyte, okay, this APC, okay, digest a pathogen, okay, for example, a macrophage, the macrophage will present the antigen on its cell surface to corresponding helper T cell. So, example, si macrophage, kinain niya yung bacteria, okay? Lahat daw ng antigen na digest niya after digestion, okay, it will be presented on the surface of the macrophage. Okay, for it to be able to present it, okay, to the corresponding helper T cell. So, the presentation is done by integrating it to the cell membrane, displaying it attached to an MHC class II molecule. Okay, so this would indicate, okay, to other WBCs that the macrophage is not pathogen, okay, despite having antigen on its surface. Okay, so we have naman basophils. So, number 24, the answer is letter D. Basophils, okay, they are capable of participating in phagocytosis, but they possess less phagocytic activity, okay, and they do not process antigens. Okay, the ineffectiveness of the cells, okay, results from very small number of cells in the circulatory system, okay, in the circulating blood, okay, and lack of powerful digestive enzyme. Okay, so basophils are generally important or functionally important in the body defense in acute, systemic, okay, and hypersensitivity reaction. Okay, in acute, systemic, okay, hypersensitivity reaction. Although phagocyte siya, but poor, okay, phagocyte. Big sabihin, hindi siya ganun ka-effective unlike your neutrophils, macrophage, and other phagocytes. Okay, we have your eosinophils. Eosinophils are capable of participating in phagocytosis, but they do not process, antigens. Like your basophils, they do not process antigens. The ineffectiveness of eosinophils, okay, results from the small numbers of the cells, okay, and lack of powerful digestive enzyme. The same with basophils. Okay, eosinophil is considered a homeostatic, regulator of inflammation. It is a regulator of inflammation. Okay, so they have a major role on host defense mechanism because of their ability to kill parasites, particularly the helminthic parasites. Okay? We have your PMNs or your polymorphonuclear neutrophils. They are particularly effective in host defense against bacterial and fungal infections. So this neutrophils, okay, they have antimicrobial function. And this antimicrobial function is very important, very essential in the innate immune system. And remember, PMNs do not process, okay, do not process antigen. Okay, do not process antigen. So number 24, the answer is letter D. Okay, letter D, macrophage. Number 25, which immunoglobulins help initiate the classical complement pathway? So discussed kanina, okay, we have classical pathway, okay, which is initiated by immune complexes, apoptotic cells, viruses, gram-negative organism or bacteria, and your C-reactive protein, which is bound to a ligand. So your classical complement pathway, it can be initiated by immune complexes. Okay, and this is achieved through which of the following immunoglobulins? So in this question, the answer is IgG and IgM. IgG and IgM. Okay, so C1 binds to the Fc region of the two closely spaced IgG, yung dalawang magkatabing IgG, okay, or one molecule of IgM. Okay, so this IgG and IgM are bound to the to an antigen, okay, expressed on the surface of the bacterium. So IgM and IgG, in particular, we have your IgG sub 3 or the subtype of IgG. Okay, these are effective activators of the classical complement pathway. Okay, so for number 25, the answer is IgG and IgM. So what are the other activators, okay, or initiators of the other pathways? So we have here the classical pathway. As seen here, this is an immunoglobulin G. Okay, immunoglobulin G. Okay, dalawang magkatabing IgG. Okay, if IgM naman, at least one molecule of IgM can initiate the classical pathway. While your lectin pathway, this is initiated when the mannose binding lectin, okay, mannose binding lectin binds to the terminal polysaccharide residues of the pathogen, okay, which is your, in this case, your bacterium. Okay, so MBL is a structurally homologous component, okay, homologous to C1q of the classical pathway. Okay, homologous, magkamukha sila. Okay, homologous to C1q in the classical pathway. We have your ficolin, although not shown here. This has structure similar to MBL and can also bind to your carbohydrates such as the N-acetyl galactosamine, N-acetylglucosamine, on the microbial surfaces. So the MBL and ficolin, they are found in the circulation complex with proteases. This proteases we call as your mannose-associated serine proteases. Okay, so once bound, okay, to bacterium, okay, this protease, one of the proteases, we have your MASP2, sequentially cleaves your C4, okay, and C2 to form C4b, okay, C4b2A. C4b2A is also formed in the classical pathway. Okay, so take note that C4b2A is also formed in the classical pathway. So it is the C3 convertase, okay, it is the C3 convertase that cleaves the next component in the pathway, which is your C3. Okay, so we have your alternative pathway. Alternative pathway, remember, are activators. Okay, it is triggered by almost any foreign substances. Okay, and in the absence of specific antibody. Thus, alternative pathway is a key part of the innate or natural immune defenses. So, the most widely studied activators include the lipopolysaccharide, okay, lipopolysaccharides from the cell wall of gram-negative bacteria. Okay. So, example of this lipopolysaccharides, okay, from the cell walls of the gram-negative bacteria, these are the endotoxins. What else? The cell walls of the yeast. Venom factor found in snakes, okay, especially yung mga cobra, no? And then, protein presence in other snakes. Okay? So, some agents that activate the classical pathway, okay, such as your viruses, okay, aggregates of immunoglobulins, and necrotic cells can also trigger alternative pathway. But particularly tayo dito, no, it can be activated through the lipopolysaccharides present in the cell wall of your gram-negative bacteria. Okay? Okay, so number 25, the answer is IgG and IgM. Okay, IgG and IgM. Ilan nga ulit molecule ng IgG? It should be, okay, 2 molecules of IgG. Okay, 2 molecules of IgG. Okay, 2 molecules of IgG closely spaced together, magkatabi, or 1 IgM molecule, okay, or 1 IgM molecule. Okay? Number 26. The curve was obtained by adding increasing amounts of soluble antigen to fixed volumes of monospecific serum. So the area showing the curve of a prozone here is, so at the x-axis of our graph, okay, so this shows the increasing concentration of antigen. At our y-axis, okay, at our y-axis, Ito po, y-axis. Y-axis. Okay? It shows the increasing amount. Okay? Increasing amount of precipitate. And remember, paano tayo nagkakaroon ng precipitate? Once there is antigen, there are what? Reaction with an antibody. Okay? Antigen-antibody reaction produces precipitates. So here, showing a bell curve graph. Okay? Tinatanong dito, asan ang prozone? As described kanina, prozone, okay, it causes false negative reaction due to antibody excess. Okay, antibody excess. So, which of the following part, okay, or which of these four sites indicated in our graph shows likely a prozone effect? Okay, dapat walang precipitate, dapat walang, okay, interaction. So, false negative dapat, or no precipitation should be observed. So, dito, increasing concentration of antigen. Antigen ba yung madami? Hindi. Okay, we have your antibody. Okay, while it is increasing here, you can see an effect, okay, or increasing precipitate. So, this would indicate letter A as your prozone. Okay, letter A is your prozone. Okay. Letter B. Ano naman yung B? So, the B area, this is the area of the curve, okay, where soluble antigen-antibody complexes have begun to form. So, letter B is, okay, in the next question, in number 27, okay, this is your letter B. Okay, meron nang napupuno or nabubuong soluble antigen-antibody complexes. In A, we assume here, okay, that we don't have, okay, precipitates present. Okay? And number 27, this is 28, this is 28. Okay, number 28. So, the curve was obtained by adding increasing amounts of a soluble antigen to fixed volumes of monospecific serum. Okay, so the area on the curve where no precipitate formed due to antigen excess, okay, antigen excess could be at which point? Letter D. Okay, letter D. Okay, no precipitate form because of antigen excess, and this is observed when there is a post-zone effect. Okay, ano naman yung C? Okay, ano naman yung C bago tayo mag-move? Okay, this is the area of the curve, okay, when we say there is a rate of equivalence. Okay, so equal parts of your serum and equal parts of your antigen, okay, producing a significant amount of precipitate. Okay? So curve showing, pwede ba gamitan natin kanina? Okay, so C, okay, shows equivalence precipitate. Okay, and the area of the curve where we observe equal amounts of antibody and antigen, therefore we can observe the production of precipitates. Okay, number 29. Okay, a technique in which soluble antigen is attached to a particle producing agglutination with a specific soluble antibody. A, passive agglutination. Okay. A, passive agglutination. B, reverse passive agglutination. C, agglutination inhibition. Or letter D, heme agglutination. Okay. So, for the Number 29, the answer is reverse passive agglutination. So let's define first. Okay, let's define first what is a passive agglutination versus reverse passive, okay, agglutination. Passive agglutination, okay, it is a technique, okay, technique in which soluble antigen is attached to a particle, okay, producing agglutination. Okay, this produce agglutination, this is a reaction with a specific, okay, with a specific soluble antibody. We have naman reverse passive agglutination. This is a technique in which antibody, okay, antibody is attached to a particle producing agglutination. Okay. Okay. So, for agglutination inhibition, ano naman yan? So, most sensitive and can detect small amounts of antigen. If antibody is present, agglutination does not occur. Okay? So, it is inhibited. Agglutination will be inhibited. Okay? So, if antibody is present, agglutination does not occur because there is always antibody bound to an antigen. While your hemagglutination test, okay, this is a assay used to detect antibodies directed against the red blood cell antigen. So this method, okay, uses antigens naturally occurring on a particle, okay, to demonstrate agglutinations. Okay, this is usually commonly observed during cross-match. Okay, the red blood cells, okay, used in cross-matching. Red blood cells in typing and cross-matching. That is an example of hemagglutination test. It is a direct agglutination testing. Okay? So, number 29. Okay, the answer is letter B, reverse passive. So, number 29, letter B, reverse passive agglutination. So, para po medyo mas naiintindihan po yung sulat. Number 30, immunoelectrophoresis involves the following. Number 1, separation of proteins based on the rate of migration of individual components in an electric field. Okay, so number 1, separation of proteins based on the rate of migration of individual components in an electrical field. 2, electrophoresis of serum only. 3, double immunodiffusion following electrophoresis. Or number 4, used to visualize the size, okay, shape, position, and homogeneity of an entire antigen spot after electrophoresis in an agar by flooding the surface of the agar with antibody. So, ano daw po doon sa mga statements yung involved in immunoelectrophoresis. So, let's define first electrophoresis. So, immunoelectrophoresis, let's define it first. So, it is under the technique of electrophoresis, but here, what we are trying to separate and identify are antigen-antibody complexes or immunoprecipitates or molecules. So, immunoelectrophoresis consists of combination of the electrophoretic step, okay, with the subsequent precipitation of an antigen-antibody complexes. So here, once the serum protein, okay, are separated in an agar gel. Okay, take note, pwede tayong gumamit ng other than serum. We can also use urine. You can also use CSF. Okay, so these serum proteins are separated in an agar gel. So this separated serum proteins, okay, these are allowed to diffuse against antibodies, leading to formation of a pattern. Okay, pwede yung pattern niya, depende kung anong klaseng type ng method. Okay, pwedeng precipitation arcs, pwedeng rocket like, okay, precipitates. Okay, so this will represent a major constituent among, okay, the serum protein. So this method is used for the detection of high antibody concentration, okay, the presence, homogeneity, and specificity of antibodies. So we have common techniques. Marami siyang techniques, but let's focuses in this two. We have your electroimmunoassay. Example is your Laurel rocket electroimmunoassay, your Laurel rocket technique. Okay, this involves antigen proteins, okay, which are transported electrophoretically through a gel slab. Okay, in a gel. Okay, this gel contains the antibodies in a certain pH level, okay, at which the antibodies remain essentially immobile or hindi sila gumagalaw. So, what will happen is that an antigen and antibody reaction will happen. Okay, so this will result in the formation of rocket-shaped, okay, precipitate peaks. Okay, a rocket-shaped precipitate peaks, okay, their height and area are linearly related to the antigen concentration. So, these techniques would allow quantitative analysis of antigens. Okay, this would allow the quantitative analysis of antigens. We have the counter-immunoelectrophoresis. Or counter-immunoelectrophoresis, this is sometimes called as your counter-current or crossed-over immunoelectrophoresis. Okay, this is done in an agarose gel of high electroendosmotic flow. Okay, so antigens are electrophoretically moving towards the uncharged antibodies. Okay, so this movement is carried by the electroendosmotic flow, okay, in a countercurrent, okay, producing precipitin arcs. Okay, producing precipitin arcs. So, and sabi nga natin, no, meron na tayong electrophoretic step, okay, and subsequent precipitation. What are the involved, okay, steps, okay, in immunoelectrophoresis, common in immunoelectrophoresis? So the correct answer in number 30 is actually 1, 3, and 4. Okay, it involves separation of proteins. Okay, separation of proteins based on the rate of migration of individual components in an electrical field. Okay, an electrical field. Number 3, double immunodiffusion, okay, following electrophoresis. Okay, double immunodiffusion, okay, following electrophoresis. Okay, this is the second step. Okay, subsequent precipitation, we have your double immunodiffusion. And number 4, used to visualize the size, the shape, position, okay, and homogeneity of an entire antigen spot after electrophoresis using agar. Okay, this is done by flooding surface of the agar with antibodies. Okay, so take note, electrophoresis is not done only or exclusively with serum. Marami po tayong pwedeng paggamitan yan. Can be in a spinal fluid, okay, it can be a CSF, okay, it can be a serum, it can be urine. Okay, so number 30, the answer po is letter C, 1, 3, and 4. Okay, 1, 3, and 4.